Hello and welcome to my channel. In today's video, we are going to discuss about hemoglobin and myoglobin structure in detail from the book Voet and Voet. First of all, I am just clarifying here that hemoglobin and myoglobin, although their roles are almost same, that they act as transporter molecule for oxygen in different parts of the body. However, myoglobin is specifically for skeletal muscle and cardiac muscle, whereas hemoglobin acts as transporter of oxygen throughout the body. These are examples of quaternary structure of proteins, immunoglobulins and hemoglobin, because both of them have more than one polypeptide chain. About hemoglobin, it is the red blood pigment, and it is one of the first proteins to have its molecular mass accurately determined. In this video, we are going to discuss about three types of hemoglobin molecules: HbA, that is adult hemoglobin; HbF, which is fetal hemoglobin; and HbS, sickle cell hemoglobin. Hemoglobin is a 65 kilodaltons heterotetramer which has α₂β₂ subunits. Myoglobin is a 18 kilodalton monomeric oxygen binding protein of skeletal and heart muscle. Hemoglobin transports oxygen from the lungs, gills, or skin of an animal to its capillaries for use in respiration. Heme is a single subunit of hemoglobin. It is responsible for characteristic red color of blood, and it is the site at which each globin monomer binds one molecule of oxygen. This is structure of a hemoglobin molecule. These are four porphyrin rings, porphyrin derivatives, also called pyrrole rings. Now you have to remember this term porphyrin or pyrrole, it comes from the chemical name of the ring. However, each subunit is called heme group, A, B, C, D. Now here what you can see is the 6 coordination ring. 1, 2, 3, 4. Fifth is oxygen, and sixth we have histidine, to which they are bound. The heterocyclic ring system of heme is a porphyrin derivative. It consists of pyrrole rings linked by methane bridges. Fe² oxidation state for the iron, the prosthetic group which is present in the center. This iron prosthetic group is the one to which the four subunits are linked. It will always remain Fe2 plus whether or not the heme is oxygenated. So whether oxygen is bound to hemoglobin or this is deoxy state of hemoglobin, iron state will always remain Fe2 plus. The Fe2 atom in deoxygenated hemoglobin and myoglobin, and 4 from porphyrin and 1 from histidine side chain of the protein. We are trying to explain here that there are two states of hemoglobin. One is the deoxy state in which no oxygen is bound, and one is the oxy state in which oxygen is bound. So in the deoxy state, hemoglobin exists in 5 coordination ring structure. 5 coordination ring means just the 5. Oxygen is not there. Just these 5. 1, 2, 3, 4 and 5. But in the oxy state of hemoglobin, it is a 6 ring structure. 1, 2, 3, 4, 5, 6. On oxygenation, the oxygen binds to Fe2 on the opposite side of the porphyrin ring from the histidine ligand, and so that the iron is coordinated. The ligands occupy 6 corners. The deoxygenated hemoglobin is dark purple and the oxygenated one is bright red. Certain molecules like carbon monoxide, nitric oxide, cyanide and H2S, they coordinate to the 6th ligand position of the Fe2 with much greater affinity than O2. The Fe2 of hemoglobin and myoglobin, they are oxidized to Fe3 and then to met hemoglobin so that oxygen cannot bind. A methemoglobin reductase enzyme is there which can convert methemoglobin back to Fe2 so that it can again bind to oxygen. So myoglobin detoxifies and synthesizes nitric oxide. Now see, basically the single unit of the hemoglobin is equivalent to the myoglobin structure. Myoglobin is just a tertiary protein. It has just one single polypeptide chain. It is a tertiary structure of protein, and whereas hemoglobin is a quaternary structure of protein with more than one polypeptide chain. Nitric oxide acts as a vasodilator, and it is highly reactive and hence toxic substance in muscles under normal oxygen concentrations. Nitric oxide is detoxified through its reaction with oxygenated myoglobin to yield nitrate ion and myoglobin. The other globins which are present are hemocyanins, hemerythrins, and let's see how oxygen binds to myoglobin. When myoglobin binds to oxygen, it forms MbO2. The O2 dissociation of myoglobin is called fractional saturation, YO2. And this is the formula for that. Now, this is a curve which shows myoglobin versus hemoglobin structure. Hemoglobin has a sigmoidal curve because it has positive cooperativity. It has 4 subunits, 4 heme subunits, and each subunit helps other subunits to bind oxygen molecule. That's why it is a cooperative binding curve. Whereas myoglobin curve having just single heme subunit, it is a hyperbola. Myoglobin's oxygen dissociation curve closely follows the hyperbolic curve. In contrast, hemoglobin's oxygen dissociation curve has a sigmoidal shape. This is called cooperative interaction. The binding of oxygen to one heme subunit increases the affinity of hemoglobin for binding additional oxygen molecules, which means if, let's say, there are 4 heme subunits, 1, 2, 3, 4, which are not bound to oxygen yet. When oxygen binds to one heme subunit, it helps other oxygen molecules to bind to other subunits. This is called cooperative interaction. Hemoglobin can bind to carbon dioxide. Hemoglobin is not only an oxygen transporter, but also plays a crucial role in carbon dioxide transport from tissues to lungs. Carbon dioxide is transported in blood in 3 forms. Dissolved carbon dioxide, 7%, carb amino hemoglobin, 23%, bicarbonate, 70%. When hemoglobin binds to oxygen at 7.4 pH, it undergoes a conformational change that makes it a slightly stronger acid. It therefore releases protons on oxygen binding and pH increases, which stimulates hemoglobin to bind to oxygen. This is called Bohr effect. CO2 and chloride, they modulate hemoglobin oxygen affinity. CO2 modulates oxygen binding directly, whereas deoxyhemoglobin can bind more CO2 than oxyhemoglobin. CO2 and H plus both can act as modulators of hemoglobin's oxygen affinity. Cl minus in the erythrocyte is greater in the venous blood than it is in the arterial blood. Cl minus is therefore also a modulator of hemoglobin's oxygen affinity. Besides CO2, Cl minus, and H plus, we also have BPG, which can affect oxygen binding affinity of hemoglobin. BPG is called 2,3-bisphosphoglycerate. This is one of the unstable intermediates of glycolytic pathway. BPG binds tightly to deoxyhemoglobin state, but only weakly to oxyhemoglobin. The presence of BPG decreases hemoglobin's oxygen affinity by keeping it in the deoxy conformation. For example, P50 of whole blood versus stripped hemoglobin is 12 to 22 times more. So BPG, CO2, H plus and Cl minus, they all 4 substances in blood account for the oxygen binding properties of hemoglobin. Now, increased BPG levels are partially responsible for high altitude adaptations. At high altitudes, rapid increase in the erythrocyte BPG concentration, the consequent decrease in O2 binding affinity increases the amount of O2 that hemoglobin unloads in the capillaries. And in fetal hemoglobin, it has a low BPG affinity. So BPG helps supply the fetus with oxygen. A fetus obtains its oxygen from the maternal circulation via placenta. Because of HbF having higher oxygen affinity than HbA, BPG facilitates oxygen binding, and HbF has alpha-2 gamma-2 composition instead of alpha-2 beta-2. It's mechanism. And hemoglobin has a deoxy T state, and also it has oxy R state. So hemoglobin exists in 2 states. T state, that is called the closed state where oxygen is not available to bind, also called tense state. And then there is an oxy state called R state, which is a relaxed state in which hemoglobin structure is open for oxygen to bind. In deoxyhemoglobin, quaternary structure interactions are constrained, and in that is the T state. When oxygen binds, it relaxes the quaternary structure, which is called the R, relaxed state. Now, the deoxy T state, in this state, the Fe2 plus ion is in a high spin state and has a relatively large ionic radius. So basically what happens, iron is bigger in size. It is outside the plane of the porphyrin ring. It's much bigger in radius than the 4 porphyrin rings. When oxygen binds, this Fe2 is pulled inside in alignment with the 4 rings. So in deoxy state, oxygen is not able to bind because Fe2 plus is much bigger in size than the rings, the heme rings. When oxygen binds, it pulls the iron atom down into the center of the porphyrin plane, forcing it to transition to a smaller low spin state. As the iron drops into the plane, it pulls the proximal histidine. Remember that histidine which we have learned that there are 5 5 and 6 coordination rings, out of which there was one histidine ligand also, which is bound to iron along with it. This shift ruptures the interchain salt bridges, holding the T state together and releasing tension and allowing the rest of the subunits to transition into the R. Hemoglobin T2R transition and cooperativity. Fe2 plus movement triggers T2R transition. In deoxyhemoglobin T state, Fe2 plus lies 0.6 Armstrong outside the porphyrin plane due to doming of the heme. Binding of oxygen pulls Fe2 plus into the porphyrin plane. And Fe2 plus movement pulls the proximal histidine and F helix, initiating the T2R conformational change. Valine E2 moves away, allowing easier oxygen binding. Changes at the alpha-1 beta-2 interface. The alpha-1 beta-2 and alpha-2 beta-1 interface, they undergo major rearrangements during oxygenation, as we discussed that they are flexible structures. Contact shifts by 6° Armstrong between T and R state, and histidine 97 beta moves from its position with threonine 41 alpha to threonine 38 alpha. These interactions act as a binary switch stabilizing either T or R states. T state is stabilized by salt bridges. Deoxy T state is stabilized by inter and intra subunit salt bridges and hydrogen bonds. And oxygen binding breaks these salt bridges, allowing transition to the R state. Energy released from the FeO2 bond formation drives this conformational change. Next is the basis of cooperative oxygen binding. Oxygen binding to one subunit. It pulls Fe2 plus into the heme plane, moves the F helix, alters the alpha-1 beta-2 interface, converts hemoglobin from low affinity to high affinity R state, and remaining subunits bind oxygen more easily, which is called positive cooperativity. Now talking about sickle cell hemoglobin, HbS. HbS is the abnormal hemoglobin responsible for sickle cell anemia. It differs from normal adult hemoglobin by a single amino acid substitution in the beta globin chain. Mutation takes place at the 6th amino acid of beta chain. Normal hemoglobin is glutamic acid, whereas the mutated one is valine. Glutamate is negatively charged and hydrophilic, whereas the mutated form, valine, is nonpolar and hydrophobic. This replacement creates a hydrophobic patch on the hemoglobin surface.